Hi everyone, I'm Michael Brenner from Brigham and Women's Hospital and Harvard Medical School in Boston, and it's my pleasure today to talk with you about fibroblasts. And I hope over the course of this session I'll help you gain a better understanding of the role fibroblasts play in rheumatoid arthritis and why we think they hold exciting promise for treating unresponsive rheumatoid arthritis. These are my disclosures. We all know what rheumatoid arthritis looks like clinically, with swelling and inflammation and ultimately joint destruction. The process, of course, is focused in the synovium, depicted here in this schematic, as a little thin membrane which completely encircles the joint and, of course, under normal conditions, provides lubrication, lubricin, mucins, and other things. Intraoperatively, this is the femoral condyle looking up at the knee, and from a patient without rheumatoid arthritis. The synovium is very hard to even find at the gross level. But below, this is a picture of the synovium in a patient with rheumatoid arthritis. You can see the femoral condyles, and then this massive hyperplastic, overgrown, inflamed tissue, which is, of course, damaging the joint significantly. Now, histologically, the synovium normally is a pretty bland, loose connective tissue. It has a very thin lining layer, just a couple of cells thick, and a sublining layer, which has blood vessels, adipocytes, and connective tissue. In RA, on the other hand, the tissue becomes markedly hyperplastic and massively infiltrated with leukocytes. The lining then is expanded moderately. The major change is the huge expansion of the sublining shown here, with a lot of neovascularization, and these kind of blue-black staining cells are all infiltrating leukocytes. And this lining-sublining difference is very important, as you'll see. Well, the topic for today is to focus on the fibroblasts in the synovium, and I thought I'd just remind you for a minute of the normal physiological functions of fibroblasts, shown here on the left. They're, of course, important for wound healing. They're important in immune responses in lymph nodes and regulation of the stem cell niche in the gut, for example. Many important functions we know well. But there are also many pathologic functions. Of course, most people think of fibroblasts and their relevance in fibrosis and scarring, which, of course, are very important. But the topics for today will be the role of fibroblasts in invasion and degradation and joint destruction in RA, and their role as inflammatory cells, something that people really hadn't appreciated so much before. Inflammation and joint destruction is mediated very substantially by fibroblasts. Well, to understand the synovium and the fibroblasts that make up the synovium is a mesenchymal tissue. So the fibroblasts actually are the parenchymal cells, the main cells of the synovium. A number of years ago, we learned that a Cadherin 11, mediates the morphogenesis of the synovium because it mediates the adhesion of the fibroblasts to one another to form the tissue. These are cell adhesion molecules shown coming from a cell above and a cell below, mediating the adhesion between the cells, in this case, the fibroblasts. And in vitro, if we culture synovial fibroblasts from a wild-type mouse, we can grow a lining-like structure in this synovial organoid in vitro. But if we use fibroblasts from Cadherin 11 deficient mice, you can see the lining structure doesn't form normally. The same is true in vivo in mice. The synovium is very hypoplastic if they lack Cadherin 11. Another feature that we found, which we hadn't expected, was that this cadherin, which is expressed just on mesenchymal cells like fibroblasts, actually determines the way the fibroblasts behave. And so if you knock out the Cadherin 11 and generate arthritis in mice, as shown here, you greatly reduce the inflammation. And if you take fibroblasts that either lack above or here below express Cadherin 11, you can see that the cells expressing Cadherin 11 are much more invasive into this matrix in this assay system. And we think this is relevant because when you look at the histology in rheumatoid arthritis, the articular cartilage is shown here. The synovial pannus, you can begin to see at these red arrows, is essentially burrowing in and eroding away the cartilage. And the cells of the pannus that are doing that are shown here below, stained with Cadherin 11 as fibroblasts compared to an isotype control. And so this is just really illustrating that it looks like the fibroblasts may be the cells that are degrading the cartilage in rheumatoid arthritis, and I'll show more that supports that idea. One of the findings from mouse studies that's a very striking study was carried out by Chris Buckley's lab, and it's shown here that if you deplete fibroblasts, you can markedly reduce inflammatory arthritis in mouse models. And that's shown here where they carried out the serum transfer arthritis model. And then here using fibroblast activation protein alpha, it's upregulated on activated fibroblasts, and a diphtheria toxin to then deplete the cells that express that, you can see that the severity of the arthritis is greatly reduced when you deplete the fibroblasts in this mouse model of inflammatory arthritis. And then remember, I commented there were lining and sublining fibroblasts, or sections of the synovium containing fibroblasts. Well, they then adoptively transferred intra-articularly into mice, lining and sublining fibroblasts that they'd sorted. And the sublining fibroblasts shown here significantly increased the inflammatory process in the arthritis, whereas the lining fibroblasts that were transferred into the joints during arthritis in mice significantly increased either their bone or cartilage damage. And so the leading fibroblasts improves rheumatoid arthritis, or the mouse models of arthritis, and it's the lining fibroblasts that drive a lot of the destruction, and the sublining fibroblasts which drive a lot of the inflammation. And that's depicted here in this schematic. The lining fibroblasts mainly causing the damage, the sublining the inflammation. Well, I'd like to mention briefly the AMP. This is the AMP RA-SLE consortium that's been going on for about 7 or 8 years, supported by NIH and company partners. And it is intended to be a single-cell disease deconstruction consortium in which biopsies of the synovium from RA or the kidney from lupus nephritis are analyzed at the single-cell level to try to figure out what the cells are and what they're doing causing the pathology. And for RA, samples are taken from biopsies at many sites around the country and in the UK. And then the cells are subjected to a pathway for single-cell RNA sequencing, shown across the top. They're also sorted for bulk RNA sequencing. And then they're also sorted for ATAC-seq, which looks at chromatin state and at immune repertoire. I'll show you just a little bit of data from this consortium relevant to the fibroblasts and our story today. Well, if you look at the single-cell RNA sequencing from the synovium, from the AMP consortium, you can ask, well, which cells are making most of the IL-6, one of the most important inflammatory cytokines in RA? And the surprise is shown here. It's this population. It's one of the populations of the fibroblasts that's the major producer of IL-6. Now, the fibroblast populations are depicted here in what's called a Tisney plot, where each of these areas in different colors represents a different cluster, a different subset of fibroblasts. And there are 4 clusters shown here. And the one that's most important for this discussion is this dark blue population. And the genes that are expressed in these populations are shown here on the right. Thi1 up here on the top is a marker for sublining. So this is a sublining fibroblast population. CD55 is a marker for lining. Those are the fibroblasts down here at the bottom. And what's important about this population, in addition to it being in the sublining, is it's the population that's expressing almost all the IL-6 and HLA-DR, which is upregulated as part of antigen presentation. So there's an inflammatory population because it's expressing all the IL-6 that's present in the RA synovium. And that population turns out to be massively expanded in rheumatoid arthritis. These are all the fibroblast populations, and this population represents 1 to 2% of the fibroblasts in rheumatoid arthritis, but this population represents over half of the fibroblasts in rheumatoid arthritis. So it's an inflammatory sublining population that's massively expanded in RA. Well, what might it be doing? When we looked histologically, and this is synovium from an immunofluorescently stained sample, you can see the synovial lining here in green is stained with lubricin, a proteoglycan 4 marker, and the sublining is stained here in blue with Thi1 or CD90 marker of the sublining. When we looked at this more closely, what we realized is that the sublining fibroblasts, they are below the lining, they're sublining, but they're very much associated with blood vessels. And we actually quantitated this, that the ratio of this sublining marker, CD90, over the lining fibroblast marker, proteoglycan 4, correlates closely with the distance the cell is away from the blood vessel. So they're blood vessel related. And this led us to do the following experiment in which we grew organoids of fibroblasts alone or in the presence of endothelial cells that organize into little blood vessel-like structures. And what we found is that when we grew fibroblasts next to blood vessels, we were able to generate the sublining and the pericyte populations, which were absent when the blood vessels were not present. And so to figure out, gee, the blood vessel seems like it's necessary to drive differentiation of the sublining fibroblasts. And remember, the sublining inflammatory fibroblasts are the most expanded in RA. What or how may blood vessels be doing that? And so we tested a lot of inflammatory cytokines and growth factors and WNTs and things shown along here in this bar graph to see if we stimulated fibroblasts with these factors, which one of them might drive the sublining phenotype. And it turned out that it was jagged1 and delta-like ligand 4. These are Notch ligands that stimulate Notch receptors, and they were driving the fibroblast phenotype. And if you ask who makes these Notch ligands, it's the endothelial cells, would come from the blood vessels. And if you ask who has the receptors for those Notch ligands, especially it's Notch 3, that's on the fibroblasts. And so this then let us ask, that's all from the human, it let us ask in the mouse, if we delete Notch 3 or use a blocking antibody to Notch 3, would that abrogate arthritis in mouse models of inflammatory arthritis? And that's what's shown here in the Notch 3 deleted mouse. Arthritis is very significantly abrogated. Okay, so we've got sublining fibroblasts, highly inflammatory, their differentiation driven by Notch signaling, and that can be important in regulating arthritis. Well, another feature that we learned about fibroblasts and their activation to produce IL-6, remember they're the major IL-6 producer in the synovium in rheumatoid arthritis, is that there's a two-step process to activate the fibroblasts. And the first is a primary signal, and the second is an amplification loop signal. And that's depicted here in this schematic fibroblast to the left. A primary signal like TNF or IL-17 activates NF-kappa B and other transcription factors that can bind to the IL-6 promoter and drive IL-6 secretion. But in addition, we found that LIF, leukemia inhibitory factor, and STAT4 were dramatically upregulated in fibroblasts when they were stimulated with several activators. And what we found is that LIF has a cell autonomous or an auto amplification loop because it's secreted by the cell and then binds to the LIF receptor on the same fibroblast. And that activates STAT4, which then binds to the IL-6 promoter, producing this massive production of IL-6. Just boatloads are produced by the fibroblast. And not just IL-6, but a larger program of inflammatory factors are released, including IL-8, CSF, CCL20, and others. And so fibroblasts are activated and then have this cell autonomous or autocrine activation loop that's important in their response. And we think this can be important from a therapeutic point of view because you can block each of the individual things that are activating a fibroblast, but you can block fibroblast activation no matter how it's activated by blocking this amplification loop. And that's depicted here. If we silence the LIF receptor for this LIF receptor axis, that blocks IL-6 production, whether the fibroblast is stimulated by TNF and IL-17 or IL-1 or LPS or any of the other things that we've tested. And so there's a large inflammatory module, prominently IL-6, but also other factors, that's regulated by this LIF receptor autocrine loop, and blocking that loop blocks activation no matter how the cells are stimulated. So I'd like to summarize some of the things we've been talking about. In RA, it's an autoimmune inflammatory disease. We have T cells and B cells and macrophages and a lot of inflammatory cytokines you know well, like TNF and IL-1 and IL-6. And what I've been highlighting is that a lot of these inflammatory factors actually work by stimulating the fibroblasts. Notch ligands are also in that group. And when they stimulate the fibroblast, there's then this autocrine amplification loop I mentioned, which takes the primary stimulus and just augments it very dramatically to get very high levels of production of inflammatory cytokines. Cadherins also modulate the behavior of fibroblasts, and the fibroblasts then have two major behaviors which are involved in the pathology of RA. The one here on the bottom is depicting the fibroblast migrating and invading and degrading the cartilage through MMPs. I've shown you some examples of that. And also its effect on inflammation by producing IL-6, IL-8, a whole variety of chemokines and cytokines. And fibroblasts are also the largest producers of rank ligand in the synovium, which of course activates osteoclasts for bone erosion. So there's many reasons for thinking fibroblasts are playing important roles in rheumatoid arthritis. Okay, well, one more aspect of fibroblast biology that's relevant to RA I'll highlight next as we begin to think about how patients with RA can be stratified. Well, stratification through molecular and cellular terms, not clinical terms in this case, has been an effort so far that is still kind of waiting for its application and its relevance, and we think that day may now be here. I'd like to summarize one example of this. It's the work from Kaz Pitsalis' lab, this particular paper, very wonderful paper last year from Revelisi. And it describes the histologic types of rheumatoid arthritis synovium that you can find, and separates them into three major groups. Some patients, when you look at the histology, and in this case we call it pathotypes, they have a very lymphoid myeloid pathotype. So that's reflected here by staining the synovium with a marker for T cells, anti-CD3, B cells, CD20, macrophages, 68, plasma cells, CD138, and a B cell marker, CD79A. And what you see is the synovium is full of T cells and B cells, macrophages, and plasma cells in this pathotype. In the diffuse myeloid pathotype, there aren't many T cells or B cells, but there are a lot of macrophages. And then in this fibroid or pauciimmune phenotype or pathotype, there aren't many T cells and B cells. There really aren't many macrophages either. There are a few macrophages. They're not so striking. But this is a phenotype mostly characterized by fibroblasts. Now, in this same paper, the Pitsalis group did this very valuable comparison of response to drug treatment and then correlated that with these cellular and molecular characteristics of the synovium. So this is the R4RA study in which they took TNF inadequate responders and treated them either with rituximab or with tocilizumab. And then if they failed either of those, then they got treated with the other one. And then you could end up with patients that either did respond to rituximab or tocilizumab, or failed response to everything. They were TNF inadequate responders and then failed response to both rituximab and tocilizumab. And they're really multi-drug non-responsive patients or refractory patients. And the reason this is very interesting is then when you look at the RNA-seq to look at the gene expression in the synovium from the biopsies that were taken prior to treatment, so this is the pre-treatment phenotype, what you find is that the non-response to multiple biologics was linked to a pre-treatment fibroblast RNA signature. In other words, these were the non-responding patients were mainly those whose synovium was characterized primarily by its fibroblastic state. And that's depicted here. These genes in red are the genes that were in the refractory or multi-drug resistant patients. And here are some of the genes: fibroblast growth factor, which is a fibroblast relevant gene; Notch genes, Notch 1 and Notch 3, which I showed you through our analyses was important in sublining differentiation; collagen genes; and Hox genes, which are also mesenchymal fibroblast genes. Interestingly, the patients who were the responders had a much more inflammatory leukocyte gene related gene expression picture. And so it's very clear that the multi-drug resistant, non-responding refractory patients really are those that have primarily a fibroblast picture in their synovium. So in summary, I've talked about the role of fibroblasts, the many pathological features they have, inflammation and tissue destruction. And then by looking at their pathobiology, we begin to identify ways that we think they might be targeted therapeutically for these refractory patients. I've mentioned Cadherin 11, which alters their pathologic behavior in vitro. I've mentioned fibroblast activation, the primary activators, TNF, IL-1, interferons. We can block one or another of these, and that works in some cases for some patients. But the problem is, if you block TNF, you still have IL-1, IL-17 interferons. You can't block everything that's activating the fibroblast. And that's why we think blocking the autocrine amplification loop I've described, such as through LIF receptor, could be an exciting way to treat block fibroblast activation, even in the face of many cytokine and other activators in vivo in the disease. And then we've also talked about the expansion of the sublining fibroblasts being important in driving a big role, a big part of the inflammation in RA, and that we could block this by blocking sublining differentiation through targeting Notch or Notch ligands. And then finally, I've mentioned the example from Chris Buckley's lab where you could deplete the inflammatory or degradative fibroblasts using, for example, an antibody, a deleting antibody targeting a marker that's preferentially expressed on fibroblasts. And so our current therapies target either the leukocytes in RA, rituximab and abatacept, block T and B cells, or the cytokines, or JAKs, which are largely cytokine receptors. But as we know, about 40% of patients don't respond to any particular biologic, and even many of them who were responding, they're not in remission, they're just getting ACR 20, ACR 50 scores. And then there's roughly 10 to 15% of patients who are refractory to all therapies so far. And so we're excited about the idea of a potential therapy in the future targeting stromal cells like fibroblasts. And that might target fibroblast activation or Notch differentiation or cadherins, or lead to selective fibroblast depletions in the synovium, any of which hold promise for treating RA in drug-resistant cases. And I'd just like to thank the members of the AMP consortium who contributed importantly to this study, a really wonderful group of people to work with and a great network and consortium. And just highlight a few people in my lab who carried out some of the work I've shown you. Kevin Way, working with Ilya Korsunsky and Shomo Rishaldri's lab, worked on the Notch story. Helena Johnson did a lot of the AMP work. Hung Nguyen did a lot of the fibroblast activation work. We've had a lot of help from Chris Buckley and his team and Andrew Filer in the UK, and a lot of help from Fan Zhang in computational studies, many of which were carried out with Shamil Rishaudri, a close and important collaborator. Thank you very much.